Welcome back, friends. Welcome back in another video tutorial from microbiology with Chagnik. So in that video tutorial we are going to discuss about another important production that we are going to do with the help of recombinant DNA technology, that is the insulin production. Okay, so in the very first video of application of recombinant DNA technology, we have discussed about the site directed mutagenesis, which is a topic of protein engineering. So today we are going to discuss about insulin. So first of all, let's discuss about a little bit about what is insulin. So insulin is a hormone which maintains the blood glucose level, and it is secreted by the beta cell of the islets of Langerhans of pancreas. Okay, so what is the function of insulin? So insulin simply maintains the blood glucose level. Okay, so first thing that I want to mention you that insulin is secreted in the form of a inactive hormone from the beta cell of the islets of Langerhans, and from that, whenever it is needed for the body, it will first convert it into a prohormone, then active heterodimeric hormone, that is the active one. So let's see how it occurs. So from the islets of Langerhans, insulin is first produced in pre-prohormone form. In pre-prohormone form, it is totally inactivated. Okay. So the structure of a pre-prohormone, as I have mentioned here, consists of a signal peptide in its N terminus to sulfide along with C chain, B chain, A chain. Okay. So the conversion of pre-prohormone into prohormone take place by the removal of a signal peptide, that is its in N terminal. Okay, which leads to the conversion of the prohormone, which is relatively active as compared to pre-prohormone. Now from prohormone, with the help of pro-convertase 1, the active heterodimeric conversion of the insulin take place. So the prohormone convertase 1 in the prohormone remove the C chain from the A chain and B chain, and as a result there is a formation of disulfide bond between the sulfur groups present here, which leads to the formation of a heterodimeric hormone known as insulin. So if we focus the structure of the active insulin, so it contains a B chain, A chain, which is joined together by two disulfide bond, and there is also another one. So that is the active form of insulin. So if we try to recapitulate what is the molecular mechanism of the conversion of pre-prohormone into active heterodimeric insulin, is that the first thing, the signal peptide will be removed from the N terminus. Then with the help of pro-convertase 1, the C chain will be removed, and as a result we will end up with a active heterodimeric hormone that consists of a B chain and A chain, which is joined together by disulfide bond. Okay, so that is what we call as active heterodimeric hormone. So it is the natural phenomenon by which insulin first synthesized from the beta cell of the pancreas, islets of Langerhans of the pancreas, in inactive form, then via signal peptide cleavage it is converted into prohormone, and then from prohormone, with the help of pro-convertase 1, the C chain is removed, and we will end up with active heterodimeric form. Okay, so this is the normal production of insulin hormone. Now my question is, why we need insulin as a drug? The answer is, there are various individuals who cannot produce insulin, or who cannot produce insulin in sufficient amount for their body, and under such circumstances their blood glucose level is not maintaining naturally. So we have to apply the insulin drug artificially from the outside, so that their blood glucose level will be maintained. So what we can do under such condition? So either we will going to add the pre-prohormone form, or we will add the active heterodimeric form. So the answer is we have to add the active heterodimeric form of insulin, because remember the drug is applied when the patient lacks the sufficient amount of insulin. So we cannot wait for the conversion of pre-pro hormone into pro hormone, then active heterodimeric form. For immediate action, we have to apply the active heterodimeric form of the insulin, so that the insulin can show its activity immediately. So we will add active heterodimeric form of the insulin by either muscular push or by intravenously. Okay. So now we have learned that what form of insulin we have to apply. Now the question is how we can artificially synthesize the compound which consists of a chain and a B chain that is joined together by disulfide linkage, right? So there can be two possible ways. One possible way is by in vitro culturing the pancreas cell and by artificially producing the insulin, then transferring them. Okay, but this process is highly costly, because in vitro culturing of a cell require a high infrastructure, a good amount of intellect, and a much amount of labor, and all this fact will reflect the cost of that drug. Okay, so as a result the drug become very costly, and usually it will be not preferable by most of the population, because most of the normal people like us cannot afford that so much expensive drug. So although the investigation, our invention and the research of the production of the insulin drug is appropriately well, but it will not work on the industrial level, because most of the people cannot afford such kind of expensive drug. So that is the main point of synthesizing any kind of drug, that not only the production is necessary, but you have to also keep in your mind that the ingredients, the time, the intellect you are using should support a well defined cost. Okay, that is affordable by the population, most of the population, so that your drug will be popularized. Okay, so we cannot use the in vitro culturing of the pancreas cell, that is not possible. So instead of that, we will going to take the help of the recombinant DNA technology and the microbial world. Let's see how. So because before going to discuss about the detailed procedure, I want to recapitulate the structure of the active heterodimeric form of insulin, that consists of a chain and a B chain which join together by sulfide linkage. Okay, and there is also another sulfide linkage. Okay, so let's see how we can create such kind of compound with the help of recombinant DNA technology. So here we are going to run two expression vector systems simultaneously. Okay, in one vector system we will insert the gene which will produce the A chain of the insulin. Okay, and in another expression vector system we will insert the insulin B subunit. Okay, along with those two things, the expression vector also contains a beta galactosidase gene. What is the utility of beta galactosidase gene? Which will be essentially beside the insulin A subunit, and in another system insulin B subunit and promoter, just adjacent to the promoter and the insulin A subunit. So what is the need to insert insulin A subunit? So the idea is the beta galactosidase gene here will act as a reporter gene. You know, reporter gene means what? Reporter gene will help you to ensure that yes, your promoter is working well. Okay, So that is the utility of the insertion of the beta galactosidase gene. Okay, that will be just beside the insulin A subunit and the promoter. Similarly, it will be in case of our second expression vector system, where we have incorporated insulin B subunit. Okay, after that we will transfer them in an appropriate host. Okay, there can be transformation procedure. For example, heat shock CaCl2 can be employed. Okay, so after the transformation, we will select them with the help of appropriate antibiotic resistant gene that we have incorporated. And also the in the culture system, we have to add either lactose or IPTG. Why? Because here we are using promoter, which is basically a lac promoter, and which require lactose or either IPTG for its induction. Okay, so we will use a culture system which contain lactose or IPTG, that will help the promoter to work, and along with the appropriate antibiotic resistant gene, which will select only those cells that have the gene of interest containing the vector. Okay, that is the idea. After that selection procedure, we will allow the host in the recombinant vector to transcribe and followed by translation. Okay, then we will end up, so which will facilitate the production of a fusion protein. Fusion protein means it will have a beta galactosidase which will attach with the A chain. Similarly, it will have beta galactosidase and B chain in the second system too. Okay, now the beta galactosidase will also help them to isolate the insulin A chain and insulin B chain from the other endogenous E. coli protein. Because remember, during the protein extraction, we will not only get the fusion protein, along with that we will also get other endogenous E. coli protein that is produced by the normal genome, that is also present inside the cell. So we have to isolate our gene of interest from those mixture of protein, right? So that can be done by the presence of the beta galactosidase, because beta galactosidase help us to separate the insulin A chain protein and insulin B chain protein via a process called affinity chromatography, where we will prepare the stationary phase with ligand that will selectively bind with the beta galactosidase. As a result, other protein will be separated, but beta galactosidase containing the insulin A, and in this system the beta galactosidase containing the insulin B will be remain in the stationary phase, then it will be separated. Okay, so now we have a fusion protein in both the cases, both the systems. Now the idea is we have to separate the insulin A and insulin B from that beta galactosidase. Okay, so for that purpose we will apply CNBR treatment, that is cyanogen bromide treatment, that will cleave the methionine residue present here, which will causes the removal of the beta galactosidase from the insulin A chain, and in that system beta galactosidase from the B chain. Now we will allow the refolding and oxidization of the cysteine residue present in A chain and B chain, which result in the formation of a disulfide bond containing A chain and B chain, which is our active conformation of the insulin. Okay, so we will start our construction with two expression vector system. That expression vector essentially contains a promoter, that is lac promoter, along with a beta galactosidase gene, which will act as a reporter gene. I have told you that what is the utility of the reporter gene. This reporter gene will ensure that yes, our promoter is working, along with the insulin gene A subunit and insulin gene B subunit in both the cases, along with the antibiotic resistant gene also. Now we will transfer them in an appropriate host via a process called heat shock CaCl2 method. Then we will select them with the help of a antibiotic resistant gene that will select only those cells that have the recombinant vector. Okay, now we will allow them to transcribe and translate, and we will end up with a fusion protein which have beta galactosidase and insulin A peptide for one cases, and beta galactosidase and insulin B peptide for another cases. Now this beta galactosidase will also help us to separate our insulin A subunit peptide and insulin B subunit peptide from the mixture of the endogenous protein of the E. coli with the help of a chromatographic technique called affinity chromatography. After that, there will be a removal process which will separate the beta galactosidase from the insulin A subunit and insulin B subunit with the help of a process called CNBR treatment. This CNBR will causes the removal of the beta galactosidase from the insulin A and insulin B. Now we will join insulin A and B, allowing the refolding and oxidization of the cysteine residue present in A chain and B chain, which facilitate the formation of disulfide bond in between A chain and B chain. Okay, and that's how a active heterodimeric insulin is constructed. Okay, so that is how easily, with the help of some conicals and with the help of a E. coli vector, E. coli host cell and a expression vector, we can construct the E. coli, we can construct the insulin drug, and that is far more less costlier than if we will try to construct our insulin in a in vitro culture system. Okay, so as a result, the drug produced by insulin, which is commonly known as humulin, becomes affordable to the population and it become popular. Okay, so let's recapitulate the entire thing that here we have discussed, that insulin drug is required for those individuals who cannot prepare the insulin by their own. So for controlling their blood glucose level, we have to inject insulin. Okay, so we have to inject the active heterodimeric form of the insulin, so we have to prepare it. So there can be two possible ways. One is the in vitro culture system, but in vitro culture system makes the process heavy costlier. As a result, the cost of the drug become, the price of the drug become very high. So instead of doing this, we will choose the insulin production with the help of recombinant DNA technology, where we are using expression vector, two expression vector and two E. coli host cells. We can prepare the A chain of insulin A and B chain of insulin A separately, then we can join them with the help of appropriate condition that will form the disulfide bond. So that's how easily insulin can be constructed with the help of recombinant DNA technology. So that is the end of today's video. So today we have discussed how insulin can be produced with the help of recombinant DNA technology. In the next video, we will going to discuss about another kind of important hormone that is nowadays produced with the help of recombinant DNA technology, that is the production of the human growth hormone. So till then, bye bye, and thank you for listening to this class.